Hello everyone and welcome to the world of Edupedia. Today we shall discuss about genetic engineering, its principles and processes. Before going into the fine mechanistic details of genetic engineering, let's have a brief introduction about genetic engineering. Genetic engineering, also called recombinant DNA technology or RDT, has now become a buzz. It is one of the core techniques that have given birth to the modern biotechnology that has enabled the use of live organisms or enzymes from organisms extensively for the production of several useful processes and products like insulin, transgenic crops, in vitro fertilization, DNA vaccines, and many more to the mankind. This technique chiefly involves changes, alteration, or formation of new combinations of heritable information into the host organism. Here, the heritable information refers to the genetic element DNA or RNA. Now the question arises, why genetic engineering? So, unlike the traditional hybridization approaches, this technique allows isolation or inclusion and subsequent multiplication of only a set of desirable genes into the host without introducing undesirable genes into the target organism. Thus, this technique overcomes this limitation and embarks greater specificity and precision. Now, from the foregoing discussion, you must be curious to know, how can one genetically modify an organism? And what are the basic steps involved? So, to your question, here lies an answer. First of all, one must precisely identify the DNA with a set of desirable genes. For instance, if you wish to produce a certain heterologous recombinant protein or an enzyme, you must look for the gene sequence for the target protein from the organism of interest. Next, what? Then you ought to introduce that foreign DNA into the host and maintain it into the host and transfer to its progeny. In fact, the advent of the construction of the first recombinant DNA emerged from the ability of linking a foreign gene encoding antibiotic resistance with a native plasmid. The two scientists, Cohen and Boer, what they did? They cut out a piece of DNA from a plasmid responsible for conferring antibiotic resistance. With this, they used molecular scissors called restriction enzymes, and then they linked this cut piece of DNA with a plasmid DNA called vectors with the help of a different set of enzymes called DNA ligases. Now, these plasmid DNAs act as vectors to carry the foreign DNA into the host organism. This way, a recombinant DNA is created. Now, when this recombinant DNA is transferred into the host organism, say for example E. coli, it replicates using the new host DNA polymerase and make multiple copies. This ability of formation of multiple copies of itself is called cloning. So, I guess by now you must have got a picture of this process. In the later slides, I shall try explain the tools of genetic engineering in detail. The process must have been clear by now. Now, let's discuss what tools are needed to accomplish this task. So, the magic wands include restriction endonucleases, also called restriction enzymes, DNA polymerases, DNA ligases, vectors, and host organisms. Let's now try understanding each tool one by one. First comes the restriction enzymes. These enzymes belong to a larger class of enzymes called nucleases. There are two types of nucleases: exonucleases that remove nucleotides from the ends of the DNA, while endonucleases remove at specific sites within the DNA. Now, these restriction endonucleases bind to the DNA and cut each of the two strands of the DNA duplex at a certain specific nucleotide sequence called the recognition site or the recognition sequence. Interestingly, this recognition sequence recognized by a restriction enzyme is a palindromic sequence in the DNA. Now, you would ask what palindromes actually are. So, commonly, palindromes are groups of letters that form the same words when read both back and forth. For example, write down Malayalam, Madam, etc. Here, in a DNA sequence, palindrome is a sequence of base pairs that reads same on the two strands when orientation of reading is kept the same. For example, the sequence GAATTC reads the same on the two strands in 5' to 3' direction. Also, the sequence is read same in 3' to 5' direction. Now, this slide depicts the different types of restriction enzymes, their cleavage sites, etc. Of these, type 2 is the commonest and widely used. As of now, you have got an idea about the recognition sites of the restriction enzymes. Now, when the restriction enzymes cut the strands of DNA a little away from the center of the palindromes, but between the same two bases on the opposite strand, the single-stranded portions are left at the ends of each strands. These overhanging stretches are called sticky ends on each strand. Why sticky? Because they form hydrogen bonds with their complementary base pairs on the cut counterparts. Here, DNA ligases aid this step. Next comes another important tool, the DNA polymerases. These enzymes aids in the amplification of gene of interest in vitro using two sets of primers, which are chemically synthesized short stretch of oligos complementary to the regions of DNA. This reaction is called polymerase chain reaction. The DNA polymerase cannot initiate synthesis on its own. Instead, it requires a priming 3' end. This 3' end of the primer has a free hydroxyl group. This hydroxyl group acts as a nucleophile, remember. The basic reaction of the DNA polymerase involves the attack of this nucleophile by the phosphoryl group of the incoming DNTP and release of the inorganic pyrophosphate. In this way, nucleotides are added in 5' to 3' direction. Now, this slide describes the action of DNA ligases. It joins two DNA molecules or fragments by forming a phosphodiester bond between the free 3' OH group and the 5' phosphoryl group of the incoming DNTP in an ATP dependent reaction. This reaction is an endergonic reaction. Now, after the formation of a recombinant DNA molecule by the action of restriction enzymes and DNA ligases, the recombinant DNA has to be delivered to a competent host cell and subsequently amplified in the host. For this, we need a cloning vector, which must include primarily an origin of replication. It is the sequence from where the replication starts, and any piece of foreign DNA when linked to this sequence would also replicate inside the host cell. Besides ORI, the vector must also contain a selectable marker that would help in identifying and eliminating non-transformants and selectively permit the growth of the transformants. For say in E. coli, the common selectable markers are antibiotic resistance genes such as ampicillin, tetracycline, kanamycin, etc. The normal E. coli cells do not carry resistance against any of these antibiotics. Apart from this, the vector must also include few, preferably single recognition site for the commonly used restriction enzymes that aid in gene cloning. And the common types of cloning vectors are plasmids, phage, cosmids, etc. In this slide, you can see a diagrammatic representation of a common plasmid PBR322. It is an E. coli cloning vector that contains several restriction sites such as EcoR1, BAMH1, SAL1, etc. and ORI, ampicillin and tetracycline resistance genes, the ROP codes for the proteins involved in the replication of the plasmid. Now, you can choose any of these organisms as a host. For the production of heterologous proteins, E. coli is rapidly, cheaply and easily grown in large quantities. Yeast has a secretory system and proteins can be secreted into the medium, hence easier to purify. With this, we summarize our lecture and give a quick view of the entire process. So you can see in the slide, here comes the foreign DNA, that is the gene of interest, and the vector DNA, say a plasmid. Same restriction enzyme cuts both foreign DNA, that is the passenger DNA, and the vector, that is the vehicle DNA, and generates sticky ends. The sticky ends are in turn joined together by DNA ligases to form a recombinant DNA molecule that is then transferred into the host organism, say E. coli, by transformation, subsequently followed by multiplication or division to transfer it to the progeny. Thank you for watching the Edupedia World. Hope to see you again.